So last week we did kind of a lecture that was a prerequisite review. Had quite a number of notes to make, and I pulled in slides from BIO220 and BIO220, which are the two prerequisite classes that most people have had. So this is the first chapter that we're going to actually start with the book. And chapter 1 just kind of gives an overview of all the other chapters in the book, kind of an overview of all of what our semester is going to take. So I kind of wanted to take us through chapter 1, through the PowerPoint, and kind of explain how the semester might be organized and kind of the things that we're going to do as we go through the semester. So that's kind of what I started the PowerPoint with. This chapter is designed to give us kind of an overview, background of genetics. We start with some vocabulary and some really kind of overarching ideas, some overarching principles of genetics. The fact that genes are inherited, the fact that genes direct protein synthesis, just some basic overall concepts. We're going to spend several weeks looking at things from the chromosome point of view, the traits and alleles, kind of Mendelian genetics. It's going to be a couple weeks before we get into the DNA structure. So last week we talked about macromolecules and kind of a review of macromolecules. We're going to do a little bit of that in this overview because what they do is they talk about how genes and chromosomes can be inherited, the fact that genes direct protein synthesis, and those are going to be the two big sections of our semester. So they first start out kind of what is genetics, kind of how do we define genetics, what is the study of genetics. Genetics is basically the study of heredity, how traits get inherited, how traits get passed from generation to generation. The scientists who study genetics are called geneticists. Yes, that's a difficult thing to say because this is a soft C sound. It kind of sounds like an S. Geneticist. Geneticists. They're the folks that study how traits or diseases get passed from generation to the next generation. This is really essential to understanding life, understanding how those traits get passed on, how that next organism is going to look and be structured. And so geneticists can be a key to understanding all of biology. So we start out with some kind of general understanding of human organisms or biology in general. That gives us a little bit of basis about what cells do, what they produce, but genetics directs all of this. So that's kind of the point that your book is starting to make in this chapter. Your book tries to define gene, and we can define gene a couple of different ways. A gene is basically a basic unit of inheritance. It's basically a DNA strand. It's basically nucleotide sequences. So there's a lot of ways that we can define genes. A gene is the basic unit that we study in genetics. It's the basic unit of how we transmit inherited traits to the next generation. It's the part of the DNA that has the instructions for making proteins. Proteins are the basis for inherited traits. But if we just look physically at what is a gene, it's just a string of the nucleotides. It's a DNA sequence. So as we go through the semester, these first two are how we will discuss genes for the first part. These second two are how we will discuss genes in the second part. Then toward the end of the semester, we'll bring in some of the population genetics and the evolutionary genetics and some of the other topics that we're going to discuss. So if we have genes, what's the genetic code? This is another term that your book is trying to define for you. So a gene is a DNA sequence. DNA is comprised of a polymer, which is a linked chain of subunits. We reviewed that a little bit last week. A nucleic acid, like DNA, is made up of chemical subunits called nucleotides. There are 4 different nucleotides in DNA: adenine, which we abbreviate A, thymine, which we abbreviate T, guanine, which we abbreviate G, and cytosine, which we abbreviate C. And so we combine these nucleotides in different arrangements to make DNA sequences. The specific sequence tells which amino acids are going to get incorporated into a protein. So the genetic code is basically how we take the nucleotides in DNA and make a protein. Translation, how we translate the code into a protein. So this is figure 1.1 in your book. It's showing you the DNA. Rather than a double helix, it has flattened out the helix to show kind of this ladder structure. It shows one strand here on the left, another strand here on the right. There's a phosphate sugar backbone that makes up the nucleotide. And then there's a base that extends to the side. That base is either an adenine, an A thymine, a T, a cytosine, C, or guanine, G. This strand is a polymer. All of these nucleotides are held together by covalent bonds. There's a second strand. These 2 strands are then held together by hydrogen bonds. That's those little dotted lines in the center. Those hydrogen bonds form between the bases. 2 hydrogen bonds form between adenine and thymine. 3 hydrogen bonds form between cytosine and guanine. You may remember from BIO220 and BIO212, and we'll cover in more detail later this semester, every 3 of these in an mRNA will form a codon and code for an amino acid. Now, most of the figures that I show you in lecture are straight from your textbook. So if they're difficult to see on the video and you have your book open, or you have your book in a different window, if you're using the ebook, you can kind of switch to a different window and look at the figure while you're listening to the video play. So you have some other options if the video isn't effective for how you're looking at the figures. Once we put these 2 strands together, we wrap them up in a double helix. And so that's the next figure that your book shows you, is the DNA molecule in a double helix. They've taken out the letters, but they're still showing you the hydrogen bonds. And now they've twisted this. And this is the structure of DNA, the way it exists in your cells. To tie this with what we talked about last week, this is the structure of the DNA when it's not replicating in S phase, and when it's not doing transcription in G1 and G2 phase. So this is what it looks like mostly during mitosis. So if a gene is a DNA sequence, what does a gene do? A gene directs the synthesis of a specific protein, a specific chain of amino acids. Genes contain nucleotides and function as the template for making an RNA copy. Because the DNA never leaves the nucleus. If the DNA ever leaves the nucleus of the cell kind of kills itself, commits cell suicide. So the DNA has to stay in the nucleus. But the RNA can leave the nuclear pores and can go into the cytoplasm. So we make an mRNA copy, and that goes to the ribosome in the cytoplasm. every 3 nucleotides is a codon. The codons in that RNA determine the sequence of amino acids in the protein. And then that protein has to fold up to have a specific shape in order to have its specific function. The gene says what the sequence of amino acids is. The gene says what amino acids to put in that protein. How many, in what order, the specific amino acids that are there. The gene says that. That's all the information that's contained in the gene. It's the sequence of amino acids that makes the function of the protein, and the protein has to be folded properly in order to function. So when a gene is turned on, it becomes a protein. The information in the gene is decoded, and it makes a protein that's going to fold into a 3-dimensional shape. So I want to talk just a little bit about protein folding. In the Blackboard course shell, under the Learning Materials tab, I have now started to organize some of the information that we're going to look at by chapter, because I thought that might be an easier way to do it. So under Chapter 1, here's some websites and some videos that are going to kind of contribute to our Chapter 1 studying. This is going to be a video that's going to take us through some folding of this particular protein. I'm going to kind of explain it as we go through in real time, and then I'm going to pause it to kind of emphasize a few things when we watch it a second time. It's only 1 minute and 6 seconds. The name of this protein, granulocyte colony stimulating factor. They show you kind of the carbon backbone structure of it, and now they fill in with letters to abbreviate the amino acids. They replace those with little balls. They're starting to show you some localized folding. This is going to be held in place by hydrogen bonds, and now the whole 3-dimensional shape of the protein can be seen. But they remind you that these are different amino acids. Each different amino acid has a different name and a different single letter abbreviation. This is what it looks like if we actually use the bond structure. And if we do a space filling model, now we can start to see where some active sites of that protein might be found. So the things that I really want to point out about the video, what they're showing you are the bonds that hold each amino acid together, as well as what the side chains are. We think about the structure of the amino acids, each has a different side chain. Some are big, some have a ring structure. Some are small, some are just a hydrogen or a carbon. And so it's these side chains that sometimes help the protein stabilize into its 3-dimensional structure. Once they show you the whole structure of the protein flattened out, they replace each amino acid with a letter. There are 22 amino acids, so they've given each one a different letter. And so they are telling you here, each of these is a different amino acid. They know the sequence of the gene that makes this protein, so they know the sequence of amino acids. So they fill in each of those amino acids. But for the purposes of folding, it's easier to see those little helixes form if they just show you each amino acid as a little ball. So then they go through and they fill in, and here we really get the idea like a strand of pearls. These are just a string of amino acids, a polymer of amino acids. So the primary level of folding for a protein is the amino acid sequence, stringing the amino acids together. Secondary structure of proteins is when these localized areas form a helix or a structure called a beta sheet. So when it forms these coils or helix, that's secondary structure. Those are usually stabilized by hydrogen bonds happening in between side chains of amino acids. It gets all the way into the final three-dimensional structure. This is tertiary folding. And then if this protein were to associate with another subunit, that would be quaternary level of protein structure, or quaternary folding. And so this figure in your textbook shows a protein, shows these little helix structures that comprise the structure of this protein. This helix structure is because of the amino acids that make up that protein, and those amino acids are in a certain sequence because of the sequence of nucleotides in the DNA. So what else can genes do? Genes can be copied. That's DNA replication. When we copy a gene, when we copy DNA, DNA gets replicated. So genes can be copied. Genes can change. They can mutate. We'll do a section on mutation. They can be expressed. Genes can be switched on and off, depending on if we need that protein at that particular time. All of your cells contain the whole genome. But your cells are not all exactly the same. Nerve cells are different from muscle cells. The difference is what genes they express. Nerve cells express different genes than muscle cells. In BIO220, we talk about differentiation. We talk about cells that can become different kinds of cells. And what causes them to become different kinds of cells is the different genes that get turned on and turned off. Genes can move. This is called recombination. We're going to get into that when we get into meiosis a little bit. Each of these events, each of these things that genes can do, we're going to look at in detail in some of the later semesters. So again, this particular chapter, they're kind of trying to give you a little bit of an overview. What is a gene? What can genes do? What is a trait? A trait is anything that you can observe, any observable property about an organism, some sort of physical property about that organism. We're going to use the term phenotype. Actions of gene products, proteins, gene products are usually proteins, produce visible traits such as hair color or eye color. They produce the structure of cells. They produce the structure of muscles and bones. They produce the things that the cells secrete. So we'll use the term phenotype as we talk about the traits or gene products. Now, one of the biggest things in this chapter is how are traits transmitted from parent to offspring. So they introduce in this chapter a guy named Gregor Mendel. Gregor Mendel was a monk. He worked in the mid-1800s looking at transmission of traits from generation to generation in pea plants and some other organisms. And that kind of forms the basis of this science that was born called genetics. And so Mendel did a lot of experiments and showed a lot of things about these heritable units. Each parent carries 2 genes that controls a trait, and each parent passes 1 copy of that gene to the offspring. With pea plants, he could do generation after generation after generation after generation. So he could see a trait and how it was passed from each of the parents through those offspring. He figured out that the pairs of these genes separate from each other. When we form sperm cells and egg cells, that they line up in a really random way, that there can be exchange of information when that happens, but that 1 copy gets into each of the egg and sperm cells, and when egg and sperm cells fuse, now you have a new gene pair. When we get into Chapter 3, we're going to spend 2 weeks on Chapter 3, because Chapter 3 really takes us through the details of some of Mendel's work. We're going to take our time to go through that. Some of that will be repeated information, especially if you had BIO220, but that's a big chapter. We're going to break it into chunks. We're just going to take our time to go through it. So if you've got a really good handle on Mendelian genetics, then you can kind of coast through those couple of weeks. But we'll take our time and go through some of these things that Mendel found. Now, Mendel had an idea of these discrete genes and chromosomes and units. It's really kind of later on that we put some of these terms onto this, because he didn't really use the term gene. He didn't really understand the idea of DNA and chromosomes when he started to use the terms gene and allele. Where do we find genes? Genes are found in the nucleus of cells. They're found on chromosomes. Chromosomes are in the nucleus of human cells and other multicellular organisms. Although single-celled organisms usually do have a chromosome, it's not in the nucleus, it's in an area called the nucleoid region. So prokaryotic organisms still have a chromosome. What we're talking about most of the time in this class when we're going to talk about chromosomes will be things like human chromosomes and animal chromosomes. Meiosis separates those chromosome pairs to make eggs and sperm. Fertilization brings them back together to create the next organism. So here's a figure from your book that kind of helps us get our head around this idea of how a gene on the DNA applies to chromosomes. They show you the DNA double helix. They start to make it look like a ribbon because they wind it around groups of proteins. These proteins are called histones, and so those proteins help to package the DNA into chromatin. Chromatin is DNA wrapped around sets of proteins. Once we have the DNA wrapped around those proteins in chromatin, that helps us really tightly package together the DNA. Lots of chromatin get wound into a chromosome. We looked at a figure last week in the PowerPoint from a different textbook, but you might want to go back and look at that PowerPoint, that shows a single chromosome, and then when it replicates, you have those sister chromatids. That's what they're showing you here. These sister chromatids, and they pull apart in mitosis and meiosis. Now this is kind of an artist's rendition. Is this really what it looks like in the cell? Yes, this is really what it looks like in the cell. This is a scanning electron micrograph of chromosomes in the nucleus of the cell. And they look kind of furry and fluffy because it's this DNA that's all wound around these proteins and wound up into these chromosomes. So chromosomes contain DNA strands. The DNA strands, segments of that, are genes. So this chapter is really starting us to try to get us really comfortable with the terminology. What's a chromosome? What's a gene? It's a DNA sequence. Now, one of the things that we can do is called a karyotype analysis. One of these is a figure from your book, and then I put a few more examples. I think this one at the top left came from your book. But what we do is we take a cell, and we burst the cell open on a glass slide, and we stain the chromosomes. We spread the cells out really thin so that we can stain the chromosomes. So they kind of look like this. Then you scan this into a computer if you're not already doing this digitally, and the computer lines up the chromosomes and pairs them up. So in humans, we have 22 pairs of our regular chromosomes, and we have 1 pair of sex chromosomes. So in all of these, they're showing you the 22 pairs, and then most examples, they try to show you a male, XY, to show you the different relative sizes of the X chromosome and the Y chromosome. This is called a karyotype. Karyotype. You can find that book in your text. A karyotype analysis is when we look at the chromosomes. Some diseases can be seen just by karyotype analysis. There's a disease called trisomy 21, where there are 3 chromosomes, 3 chromosome 21s instead of only 2 chromosome 21s. Instead of having a pair, there are 3, and that's why they call it trisomy 21. Sometimes also called Down syndrome. So this semester we're going to study 4 major areas of genetics. Geneticists study genetics. So we're going to kind of make ourselves into geneticists this semester. Again, we're going to start out, chapter 3 takes us into Mendelian genetics. We're going to spend a few weeks talking about things like genes and chromosomes and traits and alleles. And then we're going to pull in the stuff about DNA and protein synthesis as a way to really explain the mechanism for the Mendelian genetics. Now, all of that looks at genetics at just the individual level, the individual organism. What we can then do is get into some population genetics. What is the genetics like of a population as a whole? That's where we'll start to talk about allele frequencies. That can change over time. So we'll get into evolutionary genetics. How does the population change over time? How do the allele frequencies change over time? And then by the last week, we'll just kind of tie together some other types of things that we hear about in the news. We'll talk about epigenetics and those kinds of things that are in the news right now as we pull together most of what it is that we're talking about. So this is the last slide that I have in your PowerPoint, and this is kind of to give you an idea of a way to organize information. So what I've done here is I've done kind of a pretty simplistic mind map. We've talked a lot about what is a gene. That's kind of what the theme of this chapter is. What is a gene? We started out with a slide. What is it? So you can actually kind of make some spokes off of this center and write in some of the 4 things that we said genes are. What is their physical structure? We talked about the DNA. Then later we looked at a chromosome. So how are genes physically found in the cell? What do they do? We talked about how they direct protein synthesis and talked about protein folding. How are they passed from generation to generation? What other things in this chapter did we do? What other kind of other shape can we put? We talked about geneticists. How are genes studied? So anytime that we're going through a chapter, doing a mind map like this is sometimes an effective way to organize some of the information in the chapter. I kind of got you started here. You're welcome to color code. So I didn't draw any of the arrows because you might want to draw different colored arrows. And then each of these, you have space that you can kind of put some of the different things around it. I have a Pinterest board called Learning that has several links to different mind mapping websites. It has some visual examples of how mind mapping can work. But this is a pretty effective way to start to organize some information. Not only information that's in your chapter, but information that's already in your head. Because remember, we're building on a course that you've already taken. So there's already some things in your head. Some of the things that I'm saying is probably triggering some of that. You probably remember some of that. And so make sure that some of those things that you remember, that you get into your mind map too, so that you're building on the information and adding it to what you already know, so that you can kind of keep track of it on paper and in your head. I know some people are still having some issues accessing MindTap, so this week's assignment, I went ahead and just did an assignment that doesn't require MindTap. It will require you to kind of go through some of the things that we've looked at and really organize for yourself the relationship between gene, allele, chromosome, DNA, because I'm asking you to write a script. What if you were delivering an explanation to a group of students getting ready to study chromosomes? How would you help them to understand some of the information that we've gone through about what is a gene and what is a chromosome? As always, if you have any issues or questions as you're working on that, be sure and post them to the questions and answers forum on the discussion board, or send me an email, and I can post things anonymously for you. And anything else that I can do, please feel free to get in touch and let me know. Thanks.